Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. At the 2024 OHBM meeting in Seoul, Korea, we had a live podcast with an esteemed panel of luminaries addressing some of the big questions in neuroimaging. Dr. Sepideh Sadaghiani was one of the panelists. Today, she is the guest on this podcast. Dr. Sadaghiani is currently associate professor at the University of Illinois, Urbana-Champaign. She's also a faculty member at the Beckman Institute of Advanced Science and Technology. Before this position, she was trained by some truly outstanding mentors. She pursued her undergraduate work at the University of Innsbruck in Germany and the University of California, San Diego, while working with Marty Serino. Her master's degree was from the Max Planck International Research School in Tübingen, Germany, under the guidance of Uta Nopene. In 2010, she received her PhD at the International Max Planck Research School of Neural and Behavioral Sciences and Neurospin in Paris. And she was mentored by Andreas Kleinschmidt, who was an early luminary in fMRI. She performed her first postdoc at the University of California, Berkeley, under Mark Desposito, and then went on to the other side of San Francisco for a second postdoc at Stanford University under Mike Gracious, before coming to the University of Illinois, Urbana-Champaign in 2016. In this podcast, we talk in depth about her areas of expertise and some of her unique groundbreaking research on attention, EEG, simultaneous EEG, and fMRI, deriving EEG-based connectivity maps, as well as revealing unique temporal and spatial relationships between these resting state dynamics configurations and those derived using fMRI, thus opening up a new landscape to study the dynamic spatial and temporal properties of the brain. This was such a stimulating, wide-ranging, and fun discussion, and I hope you enjoy it. Well, welcome, Sepideh.
Thanks so much for having me. It's really an honor and a pleasure to be here. I've mentioned earlier, but the podcast has been really a great discovery during the pandemic for me, and I'm really excited to be part of it today.
Oh, well, thank you. It's like I was also mentioning, I'm always delighted when people come up to me and say they listen to the podcast, or they listen to it and they run or go walking or something like that. And I'm thinking, oh my gosh, that's great. Well, thanks again. So let's just start with a little bit of your history. I'd like to start out with the history. You have a really, I was looking at your CV, and your history struck me as probably one of the most impressive strings of working in laboratories and under mentors that would have given you probably like a really, really good perspective. Some of the top people in the field, everything from, when your undergraduate was in Innsbruck and also UCSD, I guess. And then you worked with Marty Serino there. Then you went to Tübingen to get your master's and Uta Nopene, and then Max Planck and Neurospin. Andreas Kleinschmidt, I actually haven't heard of. I think he's gotten more into clinical work, but he was impressive. He actually really understood fMRI at the very early stages, and his work was always creative and really good. At Berkeley, you were working with Mark Desposito, and at Stanford, you were working with Mike Gracious. That's some good training.
Yes,
and so why don't you expect? Yeah. Yeah. And so maybe just let's start a little bit before that. What was your first interest? I mean, a lot of people start with brain imaging and they get, you have a very technical, your work is extremely technically sophisticated. So a lot of people start with the technical side, they sort of take on the brain, but some people start with neuropsychology and then they become more technical. What was your first interest?
I always feel like without much effort, I just fell into a pot of gold without knowing where I was going. But my undergraduates was in cognitive science, which at the time in Osnabrück was really new and no one really understood what it was about. And they really did a great job in making it very multidisciplinary. I was just blown away thinking about the philosophy of mind. And then the next hour, I would go listen to computer linguistics, and modal logic and neurophysiology and a little bit of anatomy, just a bit of everything, cognitive neuroscience, and that was just beautiful. I really wanted to continue, was hoping to continue to be as interdisciplinary in my education. Yeah, so I did my bachelor's thesis with Marty Serino at UCSD. I just made a cold contact, 2-3 lines, and as if you know Marty,
Yeah.
you probably won't be surprised. He said
Yeah.
he didn't ask anything, who you are, what. He's like, okay, just like a single line response, like sure, and I'm like, I was young and naive. I just went ahead and bought a plane ticket, and I just showed up.
Wow.
So,
That's great.
that was my first touch on fMRI, and Marty's incredibly methodologically sophisticated. I mean, he was decades ahead of his time. He had surface models before the bolt signal was basically
Yes.
a thing.
Yes.
So yeah, I think it was just a bit too early for people. A lot of people just don't appreciate
Yeah.
the
Yeah.
sophistication. And I had no idea. I showed up there and I was doing statistics in surface space, not in volume space. When I came back to the real world and I started talking to people, they're like, we don't understand the pictures you're showing us. I'm like, how do you not understand it? It's the brain in 3 dimensions. They're like, we don't know where in the brain you are. We need the slices. And I'm like, wow. Okay, so not everybody thinks the way Marty thinks. So I think his undergrads has some like geography or something in it. He always thinks in terms of maps.
Yeah.
And that's
Yeah.
just his mindset. It's just beautiful. And so that was my first touch on fMRI and really seeing how he lit up like children when they get a new toy. Like when he was looking at data, that's how Marty is.
Yeah.
And so, that really inspired me to look into graduate programs that pursue cognitive neuroscience specifically.
Okay. Okay.
And so I applied to the Max Planck Graduate School in Tübingen, and that includes some rotations and looking at different labs and interacting with different supervisors, and that was really great. It's a really high quality education, and I learned an amazing amount, including at the time Camille Uledag was there at the brief moment where Camille Ugobil was at Tübingen. So there was a slight
Oh
moment of
yeah.
overlap
Yes.
in
Yes.
which I really started thinking about the bolt signal at the level of the constituent signals that create it, which is more than most cognitive neuroscientists, unfortunately, get the luck to do. So the degree of methodological thinking, I was just barely trying to hang on, as a late,
Yeah.
undergrad, early grad student,
Did
and
you?
so that was really helpful.
Yeah. Did you talk? I mean, Nikos Logothetis was there at the time too.
Absolutely, yes. And so I didn't work with Nikos. He obviously his presence was always very impactful, even when he was not in the room.
Yes.
I mean, I would see him, at like rare social occasions, but the people that worked with him obviously had a great impact on my general understanding at the time as well. So at the time, I wasn't thinking about electrophysiology and fMRI, although I did make a rotation during that master's program at Rainer Goebel's lab in Maastricht.
Okay. Okay.
And thinking about concurrent imaging, obviously we didn't actually do it because it was just a 2 month visit, but I was hoping to do multimodal work very early on,
Okay.
although I never got involved with Nikos's work at the time, and I didn't have enough appreciation of it until way later. I did my master's thesis with Uta Nopanai, and she's really interesting because for me, reflecting back, I remember some of the things that she told me that are still in my mind. Like we have a junior science paper on multimodal multisensory integration, and she was saying, "Oh, the statistics you're doing, your contrasts are actually a three-way interaction, but we're never going to tell the world that. We're not going to write it that way in the paper. You're going to say you did your two-way interactions in these three different conditions, and you compared them." And I'm like, her mind is so, the sense of like multifactorial approach to cognition, very, very sophisticated. And when I then moved to Andreas' lab, at Neurospin in the Greater Paris area.
Yeah.
It's so interesting. I think in cognitive design, he's basically the opposite of Uta. He always told me a good horse never jumps higher than it needs to. So if you look at our work, like the experimental designs are so incredibly simple. You could not possibly imagine answering the same question with anything simpler than that.
Yes.
So basically, they got both of these approaches to cognitive neuroscience. The actual reason I applied to Paris was that I was perhaps young and naive, and I thought, okay, what do I want to study? I want to solve consciousness. I want to study consciousness.
Oh, we all do. We all sort of have gotten into this.
And so I kind of looked up the people. I'm like, this group that the larger INSERM unit under Stan Dehaene has all of these different PIs that all do these really exciting types of work. And so I just reached out to Stan, and I, flew over there and gave a little talk. And so I just connected with Andreas, and then, that's what happened. I mean, we ended up studying perceptual awareness in a very narrow sense, but, you can't really solve consciousness in the MRI scanner.
Yeah,
I didn't know that at the time. But yeah, I completed my research with Andreas and actually defended back at the Max Planck graduate school. So it was sort of this international setup. And then I applied to Mark Esposito's lab. I mean, not much needs to be said in terms of his impact on the cognitive neuroscience, neuroimaging community. What's interesting about Mark's lab is how broad the topics are. He never, makes us study anything that's in his mind. It feels like everybody who comes to his lab somehow comes with some idea that may or may not be related to what's going on in the lab. And he's like, okay, sure, go ahead and do it. And so his mentorship style is also very different, but also so effective. I mean, it is just incredible when I compare these chain of people and, how much I appreciate what I've learned from them and how different that has been. So that's been, it's been incredible.
Yeah. And then at Stanford, Mike Gracious as well. I mean, he was
absolutely also, again, very different. Just the fact of moving from Berkeley to Stanford was pretty eye-opening. Such a different approach to, neuroscience, and also just physically, where, who interacts with whom, and the fact that Mike was actually more part of neurology in the med school than the psychologies, right? And having those connections. Mike at the time had been already on a route that has now continued, which is doing more and more genetics. He started, it feels like he was in genetics neuroimaging, and now the imaging is gone and it's just more and more genetics, it feels. But I had a very short stay at his lab, about 9 months or so, but I learned so much about molecular genetics. I in my naive mind, I thought genetics is simple. Once you look at the alleles, there's black or white. You either do or do not have this combination of alleles.
Yes.
But oh my God, was my eye. I mean, I was just blown away by the journal clubs and the discussions, and he wanted me to brainstorm and come up with something I would do in that realm. And it really, opened the direction that I've continued in some other ways more recently. So we've looked at heritability in traditional twin designs, but looking at EEG connectomes.
Yes.
And comparing the effect sizes with fMRI, which is entirely mind-blowing to me.
Yeah.
In fact, just this morning, there was a paper accepted, so I can mention it, on the molecular genetics of of the fMRI connectome dynamics. So,
Okay. Oh, wow.
yeah, I mean, it's on that edge of, select gene studies, which are, very hard to develop in a way that's I mean, the molecular genetics has gone so much in the GWAS direction, and it can therefore only answer very limited types of questions with very limited types of assumptions about linearity of the alleles. It's just an
Right.
additive model on a gigantic population and can only get at very large effect sizes, which
Yeah.
don't really work in the general healthy population. And
Right.
so I think there is something lost when we step away and say, select gene studies are not allowed, as opposed
Yeah.
to saying, okay, if you pre-register them in the right way and you look and you think about your power and the effect sizes, there may be a route, at least, to say, here is an effect, let's see if it replicates somewhere else, right?
Yeah.
Let's put it out there and say, yes, we're never 100% sure with a single publication, no matter what the method is. But anyway, that's all coming from Mike Gracious type of thinking to try and bring these worlds together. And
Interesting.
yeah.
Interesting. Yeah, I don't know. I mean, obviously there's Paul Thompson who has who combines his genomics with his large databases. Allen Braid Institute is kind of useful data space for understanding that as well. Yeah, I always find that when I read at least the way the genetics research is done, they might say, oh, there's they show these Manhattan plots, and then they show like some peaks, and they say, well, this explains 10% of this, and I don't even know how to think of that. It's like
That's right.
I'd like to think in terms of mechanisms or something like that. So yeah,
Yes.
um, but that's interesting. I mean, I think that there'll be more work along those lines. And in fact, I was going to talk maybe at the end, towards the end, your recent paper. I thought that you were talking about heritable traits and non-heritable traits and how they relate to the EEG connectome. And actually, some you said, oh, there's some that are non-heritable, which means that it's or it was related to intelligence, I guess. And
That's right. That's right.
yeah, and there's some that are heritable, and there's some that it was intriguing that you even mentioned that, well, some are not. Some look like they're not, and that seems like then it's subject to plasticity or something.
Right. So these recent papers in network neuroscience, there's two of them. One is focusing on the heritability of the EEG-derived connectome dynamics. And then the other one in the same data set is looking at the association between the heritable EEG connectome dynamics and a cognitive battery, right? So
Okay.
these are two separate parts that go together. And
Yeah.
so what we did is we essentially applied HMMs to source space EEG. And the source signals, were more used in fMRI to applications of data-driven state definitions, such as with HMMs. And,
Yeah.
you force them to be discrete, which of course is only an approximation. But if you look at that, the different EEG frequency bands result in different speeds of state transitions, which is also kind of interesting. But a lot of people are very hesitant about using EEG source space data for connectomics. And I was certainly one of them for a very long time.
Yeah.
I tell you in a moment, perhaps, how I got out of it by hiring a postdoc who completely blew my mind and convinced me otherwise, convinced me that I was wrong being hesitant to do connectivity in scalp EEG. But about the heritability study, so the HMM-derived connectome states, the model gives you the proportion that each state occupies of the total recording, and it gives you the probabilities of any sets of states to transition into each other. And these are different from stochastic.
Yep.
And so we find things that people have done for a longer time in fMRI. And so what's interesting is when we look at large twin data sets, and we were really lucky to have access to the Minnesota twin family study data set, we had about 1,000 twins.
Okay.
And so this is they've been followed since the 90s. Some of these, it's quite an incredible data set,
Wow.
really.
Wow.
So we have their young adult data time points at resting state, just a few minutes of EEG recording. And so when we look at a traditional twin approach, so looking at the monozygotic twins and the dizygotic twins, And then people who are in that data set, but where we don't have the twin data, so we just pair them together by sex and age matched pairs. So we have
Yeah.
unrelated, we have siblings, and the non-identical siblings, and then we have the identical twins. And so if you just look at the similarity of the transition probabilities and the similarity of what states are more prevalent than other states, it's incredibly heritable. In fact, it's more heritable than we found in the HCP fMRI data, which is just mind-blowing.
That's interesting.
So, for me, this is really strong evidence for saying that EEG connectomics is actually informative. It's not just artifacts. And you mentioned about the parts that are not heritable. I think that's important to mention, because one might say, well, genetically more similar people have more similar anatomy, so their source reconstruction could bias your similarity estimates, or something else about being twins is artifactually generating similarity about the dynamics. But what's interesting is if you look at the actual spatial pattern of the individual states,
Yeah.
and you either look at specifically functional connectivity edgewise, or you look at global graph measures such as modularity, they are not more similar among twins. So the spatial way these states look is
Yeah.
not genetic, at least in this data set, is not genetically determined.
Yes.
Whereas the way they switch and how much time they occupy, that's what's heritable.
Oh, that's really interesting.
So it is the temporal way the brain moves through the connectome
Yeah.
space, if we want to simplify it to a finite space, right?
Yes.
That's what's most heritable. So that was really, really surprising to us. And in fact, the reason I'm doing more and more EEG connectomics is findings like this, because if I care about information that is related to cognitive traits, that was the second paper. So these same heritable dynamic properties are actually related to certain factors of the cognitive battery,
Yeah.
mostly over tasks that require executive functions. So
Yep.
when I think about that, then I'm wondering, this is a cheap method. This is a method that's already in the neurology. Doctors know how to use it. We can give them a higher coverage, and we can give them software to source localize, right?
Yeah. Yeah.
Can we imagine stepping away from fMRI when we think about perhaps something that would actually translate? I'm not saying that we're close in EEG any closer than we are in fMRI. Again, you can find group results. And when were we at the point, if at all, to
Yeah.
inform a neurologist to do more with their data than what they're currently doing, right? So they are looking at the EEG spectral properties in a very global sense.
Yes.
But there could be more if we just allow this method to do more than what we've done with it so far.
Yeah. I mean, it's so easy to collect the data. I mean, the EEG is easier than fMRI in that regard. It's cheap, it's mobile, and you're right. It's sort of like, there's more, and what you're telling us is there's much more information in the EEG signal. I mean, people usually just break it into the bands and they say, oh, well, alpha, beta, gamma, whatever. But you're right. Looking at things like you said, like transitions or dwell times or patterns or sequences, who knows, of the connectome states could be informative. And so it's interesting. I was actually, when I was reading the paper, so the idea was that you said that the transition. So that's interesting. The transitions, or the transition, how they transition is related to intelligence, but oh, I'm sorry, yeah, the heritable traits and the where, but the actual configurations that are transitioned to are sort of independent. They're not.
Yes, at least in our hands, with our approach. Of
Yeah,
course, we tried a variety of spatial features, and none of them was heritable. And so,
yeah.
so we tried our best, and then we didn't find anything that
So when
was.
you were talking about intelligence, so is the idea that, so I saw that you had a pie chart, and you had
Mhm.
what was most related, and was it like, so the idea that if with intelligence is it more rapid transitions? Is there more? I mean, is it like I mean, when you talk about the probability of transition, I mean, I actually look at it the way
Yes.
I think of it. It seems like it's related to the dwell time in some sense, or how long you're in a state. I
Mhm.
mean, if you're
Mhm.
in states less long, then you would have a more light
That's a good
layer.
question. So one, of course, one difficulty with applying CCAs or any multivariate, associations and having these significant modes is that you then need to map back to where they came from, which we did. But then, on the EEG side, we took principal components of the various different temporal features that we had from the
Okay.
different EEG bands. So we put them all into one PCA and take that out. So we would have to trace back a few more steps to try and answer your question. We did not look at the average dwell time. Interestingly, we think that it's the pattern over all transitions, not, if you have you have 4 states, you will have a 4x4 transition matrix. And it's really the combination of all of that information that I think drives the cognitive aspect. But you're right that we should actually look in more detail and see if we can understand some of the more mechanistic explanations. So what is, if we split the participants and we take the top and bottom quartile performers, is there a specific pattern in their transitions that emerges? I would assume it is, and that's a great idea.
Yeah, it's interesting to then, look, right, fMRI is going into like individual differences and trying to sort of parse out the mechanisms behind that. And so, but people, I think, listening to this now would be, I think those who work with the EEG think, okay, it's such a noisy signal. How do you, they can barely extract, alpha waves or whatever. And so how do you do this? I mean,
it's very, very noisy. And also leakage correction is a terrible thing that we have to do to our data.
Yeah.
We always lose information that I'm very convinced is actually neuronal in nature.
Yes.
But in order to convince ourselves that we're not looking at leakage alone, we have to somehow remove it. So often in our papers, we do both with and without, and we report both and say, hey, the effect is still there after correction, but it may not be as strong, because zero lag connectivity actually is a real mechanism in the brain. And if you just remove it because you're worried it's created by leakage, then you are removing information. You're right, it's incredibly noisy. And it is so much more noisy when you record it in the MR scanner, even after artifact rejection.
Yes.
So often people talk to me and say, hey, I'm thinking about this question and maybe doing it multimodal. And I say, okay, come on over to my office. We're going to have a conversation. Usually, by the time they walk out, they're like, I'm not going to do this. I'm not going to do this concurrently. So let me show you the data, okay? If you give me a really good reason why you want to destroy your EEG to this degree, and that reason is more important than having good signal and do the experiment twice at separate times, then go for it. But otherwise, there is a limited set of questions that really benefit from concurrent imaging. And I really want more people to do concurrent research. It's just that, I don't want to, once you see the publications, it all looks nice and neat, right? I just want people to realize the price you pay in terms of signal to noise for a methodology that is already quite noisy. That said, fMRI is also very noisy. And
it's also very noisy as well. And yeah, we struggle with it. And as you're right, putting, it definitely affects the gradients and everything like that. And also, and then maybe it might lend itself to, so you mostly look at resting state as far as this is concerned, it looks like, but you could have somehow a naturalistic stimuli or something. And then you could do that inside and outside the scanner as long as it's time locked or something, but it's still not quite the same. But yeah, there's so much more to be done with that. And well, actually, when in the paper that was, so let me just back up a little bit. Okay, so let's just, I got way ahead of myself, but, but this is great. So how did you, so one question was on my mind. So what system are you using? I know that everyone's worried, concerned about, there's only a few systems that actually work well in the scanner. Yeah, I'm just kind of curious what.
That's right. So we're using brain products, and the main reason is that you can put it inside the MR bore. Yep. So I'm not aware of other systems that are allowed. They're allowed in the MR room, but not inside the bore. And where the biggest artifacts come from, I mean, some of the biggest artifacts is between where the electrode leaves the head and where it hits the amplifier.
Yes.
And so that lead, I mean, we put it on a sled, we sandbag the hell out of it. And, if you imagine there is even more that needs to go around before you even reach the amplifier, oh my goodness. I mean, We've been very happy with that system also for the customer service side of things. So really Brain Products has been doing, in our experience, a great job making sure that there is someone we can talk to when we need to change our setup or have other questions or issues with our caps or anything else. So that's a system we're using. You need at least 64 channel system to do any source localization.
Yeah.
I don't think going much higher density is actually in our advantage. I don't, just judged by the literature, it does not seem to improve source information that much. And if you want to do relatively long recordings, you want to go with gel rather than saline solution systems. And so with gels, the capping already takes an incredibly long time. And
Yeah.
so that's what we settled on. We have a 64 channel system from Brain Products. And
Okay.
there isn't really a whole lot of decisions you can make in terms of the montage. They may allow 3 drop downs. Usually they are happier if you only have 2
Okay. Okay.
for safety reasons.
Yeah.
And so, yeah, that's
Okay.
what I've been working with.
All right. All right. So you just started at Urbana-Champaign, what, in 2022? Yeah. And,
Well,
or
2016 actually.
2016,
So
that's
I've
right.
been
That's
here
right.
for a
You
while. Yeah,
you became
that's,
right, that's right. You became an associate professor in
that's right.
2022.
That's right.
That's right. So you started in 2016. And so and it's funny because I always people are sort of have blind spots to sort of like, they look at the East Coast and the West Coast and all the big centers and they don't really think and actually I've been following, like, for instance, I mean, there's a lot of people I know at the Urbana-Champaign. There was one guy, Art Kramer, who was there, who sort of pioneered plasticity and learning, and
Absolutely. Yes.
it's a great place. And so, what's your impression? How is the environment? I mean,
Oh, I can see myself. I cannot see myself moving away anytime.
yeah,
I mean, where would I go? I know this is a hard thing to say for someone who moved here from the Bay Area. I have to
yeah,
say, who loves water sports. I'm as landlocked as you can make it, and it's as flat as you can make it here. So, um, but work-wise, when I just had no idea how important environment was when I accepted the job. Usually you're happy to get a job and you're like, oh my God, this is great.
yeah,
But you don't realize, I mean, I didn't realize until years later, not just in terms of the equipment. So my lab is inside the Beckman Institute, and I can tell you about all the amazing infrastructure that the Institute brings, including the 2 Prisma scanners and more recently the 7 Tesla Terra.
yes,
But the really what makes the difference for me is the colleagues. I mean, from the beginning when I came here, the senior colleagues basically every time we met or talked or emailed had the attitude, what do you need? How can I make this work for you?
wow, that's great.
And it has even continued now, since I have tenure. I think I was expecting this would stop at some point, but it's just like the other thing is people are, it's all about collaboration and not just on paper. So I've been doing a little bit of soul searching now after tenure. What is it I really want to do? It's back to consciousness,
Yeah, yeah, you can do
So
whatever you want now. Yeah,
go to the big questions. And so I've started some collaborations in MRI engineering, and I know nothing about MRI, right? So um, the experts didn't push back and say, oh, you're naive. I mean, why would you? This is not going to go anywhere. They said, oh, yeah, come on, let's talk. Let's do this, let's collect some data and see what happens. And so
yeah,
that attitude has been continuing, and I'm really enjoying it because I'm allowed to be thinking out of the box, even in places that I'm very uncomfortable.
yeah,
I know someone is comfortable, and so when I go to that someone, this far, people have been incredibly supportive, and it's just blown my mind. I mean, I would never think this deeply about the almost quantum level basics of
yeah,
MRI had I not been at this place.
yeah,
It feels like you need to understand some basics about the different imaging sequences as a cognitive neuroscientist. But if you really want to ask deeper questions, which I've been kind of fallen into, given that I've looked at the relationship between electrophysiology and the fMRI signal, I feel like I really need to go deeper. But I mean, I'm not a quantum physicist, and I'm certainly not an engineer. But I have people I can talk
Yeah.
about these things with, and it's been really a pleasure
Yeah.
doing that. Whether we get any funding at some external funding at some point is another question. But
And yeah, I
yeah,
mean, the tradition with MRI, I mean, it was started with, Paul Lauterbur did all his great work there. And
absolutely.
yeah, my former postdoc, Yahui Chai, is now sort of heading up the seventh, and he's super smart. And,
Absolutely, yes. Very excited to have him here, and the community around the Seven Tesla is growing. And yeah, I mean, the cognitive neuroscience community is also growing, and I'm very excited about all the possibilities of collaboration.
yeah, and I was really struck by that. I mean, when I followed, I'm still intrigued by the whole, brain plasticity work that Art Kramer kind of pioneered, and I was really struck by, I talked to him a few times, and how it appeared that it was very high level, but very collaborative, and maybe it's the Midwest influence or something like that. It's like very, yeah,
There's definitely something about the Midwest. And it's
yeah,
so interesting if you look at a lot of the people who are here, unlike me, they have been here somehow at some point of their career, or they have some contact with the Midwest, and they come back because they can appreciate it. I had no idea. I was like like you mentioned earlier, right?
yeah.
I was the person that you were describing in terms of, oh, there's the West Coast and there's the East
Right.
Coast, and there's this big swath in between. They could have built the campus slightly closer to Chicago. I would not have minded that, but it's okay. It's doable.
Yeah, but it's a cool campus. It's all part of the Big Ten sort of
Absolutely. It's
system, and
huge, and it has basically a little bit of everything. I just by I don't know what kind of coincidence I fell into a large scale NSF 7-year project with 15 investigators,
oh wow,
which is, it's just mind blowing the kind of people I'm in contact with. I've never looked at tissue level neuroscience before, and integrating that with electronics and things like that. There is a lot of very unusual kind of out of the box research going on, and it seems like no matter what you want to do, the person of it is somewhere on campus. You just need to go find them.
yes,
So
yeah,
it's
that's great. That's great. Yeah, so let's go back into your. I wanted to sort of cover briefly your earlier work. I mean, if you call it earlier, it's from maybe 2016 or something like that. When you were working with Mark Desposito, a paper struck me as interesting. Sort of looking along the lines of, so this was from cerebral cortex, functional characteristics of the characterization of the singular opercular network in the maintenance of tonic alertness. And so, it seems like the term, attention has been sort of defined and further refined in some sense, where there's, phasic attention, there's arousal, and they're all sort of linked in some way. And it was interesting how, you had a nice experimental design that sort of pulled this apart in some sense. And it seems like that also is intrinsically related to conscious awareness, of course. Just looking at your paper, I mean, the first question that came to mind was, what do you think is actually the various mechanisms of what's going on? Is it, you can find these attentional networks, but then for arousal, but then there's phasic attention, and uh, uh, if you were looking at sort of the whole time course of, when you direct your attention versus move it, can you pull those apart just by looking at the time course, or do you? And it was also intriguing. And actually, I don't know if this was this paper, but you had another paper which I did not know, and it just shows that there's so much literature that the alpha frequencies are, basically are inhibitory in that sense. And so maybe the spotlight of attention basically inhibits alpha frequencies to the area of cortex that, then activates them or allows them to become more activated. I don't know how that ties in, but anyway, I'm just sort of speaking.
Yeah, they're all tied together. You're absolutely right that the paper you mentioned, which was fMRI with Mark Desposito, ties into the concurrent imaging with a focus on alpha oscillations that I've done separately. And the story all comes together. And we wrote a trends in cognitive sciences
Yes.
paper just to pull it all together, these different strands. But one thing about what we used to call the tonic alertness network. I've reverted to anatomy now because there's too many labels, too many functional labels for networks. And so really what we're talking about is the singular opercular slash somewhat salience-ish network. I know in some atlases they're not one and the same and they're anatomically dissociable. When I say single opercular, I mean, no dissociation, kind of a combination of all of the above. Because in this experiment and other experiments we've looked at, they have behaved the same way. So that's why I don't dissociate them. When I do, I find the same findings. So this network that comprises dorsal anterior cingulate cortex, parts of the thalamus, and parts of the prefrontal cortex, the anterior insula, or wrapping into the operculum as well. It's if you look at main effects of any task, even the simplest task you can imagine, the simplest, press a button when you hear a beep, don't press when you don't hear it. Okay.
Yep.
You see this network. I mean, I haven't in my experience seen any task versus passive baseline where this network was not there.
Yes.
So for me, it is the first basis to do anything cognitively. It's got to be there. It is to disengage from unnecessary processes and be there. It is the be there network. You can call it vigilance. You can call it tonic alertness. You can call it tonic attention. Whatever label you put on it, it is the yes, I'm here. I'm ready to do something from whatever else it was that I was doing.
Yes. Yeah.
And
orienting
so,
or something. Yeah.
Right. It's the deliberate being present kind of network, in my mind anyway. But on top of that, you need to build much more selective processes. It is the specific things you want to do. If you're looking for your red purse, or you want to hear someone's voice among other voices in a cocktail party, or anything that is specific and therefore primarily processed by a dedicated neural population somewhere in the brain, you will need a spatially more confined filter, a focus, a spotlight of attention. And I like to call that selective attention to dissociate it from this tonic type of being presentness, which is, in the experience of my results, not selective.
Yeah. Okay.
It is universal. It's the baseline. And then, so this universal tonic availability is, as far as I've seen in our research, in the singular opercular network, whereas selective attention, at least in the visuospatial space, but also auditory spatial, I mean, as soon as space comes in, perhaps also other selective features, although we haven't directly probed it, involves the dorsal attention system.
Okay.
And so that builds the next layer of selectivity on top.
Okay.
And then there is another aspect, which is the lateral frontoparietal network, the lateral dorsal prefrontal cortex, and parts of the parietal cortex, which people like to call executive control network or similar. What we have seen is that this network might be more involved with making the right regions of the brain communicate with each other that are necessary. It facilitates communication selectively between certain brain regions. This is a very simplified model, and it's based on limited amount of data. But when we tried to bring these different experiments that you mentioned together, make sense of them, that's the picture we came up in terms of the network. But what's interesting about these three networks that I named, which are the primary so-called task-activated, task-positive networks that we see in cognitive neuroscience literature, these three networks all have a very unique relationship to the alpha oscillation band, which they don't have to other frequency bands. And so when you look at spontaneous fluctuations of the power of alpha always goes up and down, even if you keep your eyes closed. So that's different from the eyes opening or eyes closing effect of alpha. If you just look at the spontaneous changes in the power of alpha, it is correlated positively with the fMRI signal in the singular opercular network. So when alpha is strong, the singular opercular network is strong.
Interesting. Okay.
But it is negatively correlated to the fMRI signal in the dorsal attention system.
Yes. Okay. Okay.
So, and I'll come to the frontoparietal because that's a separate story. But the way I see this is there is a lot of literature showing that the alpha oscillation corresponds to a rhythmic inhibition of processing in the cortex, which also translates to rhythmic inhibition of perceptual abilities.
Okay.
It really is a rhythmic inhibitory signal of some sort. And Andreas Kleinschmidt gave me the best analogy I've ever heard for it. He calls it the windshield wiper of the brain. Because what's interesting about it is it's not just shutting down.
Yeah.
It's clearing and allowing new information to come in. It's a reset button. It's a rhythmic reset button. So the brain can organize itself relative to that rhythm so that not everything disappears. You just want to you don't want to massively just suppress stuff, right? You want to have you want to have timing information about when things are suppressed and new information can come in.
Yes.
So to me, the positive correlation of the singular percolate network with this rhythmic inhibition suggests that the ability to be present is mechanistically defined in the brain or implemented in the brain as a rhythmic windshield wipering.
Okay.
And so that allows new information to pop in.
Yeah.
But now you need the dorsal attention system and perhaps the frontal parietal and other top-down control systems to actually now define what is supposed to come in and how is it supposed to be processed.
Yes.
So if you have now a relatively spatially widespread increase in this alpha windshield wiper, you would have the dorsal attention system come in and in a center surround mechanism, like think of it as a Mexican hat, you would have the dorsal attention system come in and very selectively for some specialized neural population or brain region, reduce this wiping.
Yeah.
Yes. Enhance the gain of processing in that region relative to the rest.
Yes.
So for me to have selective spotlight, you actually need the global increase in alpha. That's your baseline. And then you can selectively release that wiping. And that's what the dorsal attention system could do, which is why it's negatively related to the power of alpha.
Okay. That's actually a really nice explanation. I like that.
So again, it's a working model, right? I'm sure it's not going to be exactly correct, but it's where we are at the moment with our thinking. And then the front parietal system, what's interesting is in the same EEG fMRI data set that we observed this positive and negative relationship to the alpha power, within the same data set, if you look at the spontaneous increase and decrease of phase locking, coherence, phase coherence of the alpha cycle, you don't find either of these two networks. You find the lateral frontoparietal network.
Huh.
So when the fMRI signal is high in the lateral frontoparietal network, the phase coherence of alpha across the different areas of the brain is going up.
Interesting.
And that's independent of power, which is totally mind-blowing. It seems to be a different mechanism. It's in the same data set. We just have 3 different regressors, right? One EEG regressor is alpha power. Well, actually, we have 2 regressors. One is alpha power, and you look at the negative and positive fMRI mapping to that. And then the other regressor is simply coherence of the alpha band. And so that's how we came up with the intuition that the frontoparietal network perhaps guides a little bit what in the brain is synchronizing with other brain areas, in the sense that this wiper now needs to be aligned. It doesn't have to be fully zero lag, right? It can be off, but as long as it's staying consistent, then these 2 brain regions have some way of sending packages to each other and receiving.
Oh, that's really good. That's really interesting. Yes. Yeah. Okay.
So anyway, that's how we were trying to understand, as you said, these larger top-down control functions or attentional functions defined by physiological mechanisms as opposed to defined by psychological observations, even though I think they actually beautifully map.
Yeah.
You can come from the Posner view of attentional functions, and it would still map to this physiologically defined mechanism. Again, it's totally simplistic. I haven't talked about any other frequency in the brain, which obviously has a lot of functions.
Yeah.
But just in this limited view, this is, the current model that we have.
That's really okay. So that's actually really interesting. So once again, the alpha frequency is, so it's at the rhythm of the alpha frequency that it does this clearing, but then of course attention waxes and wanes in some sense. And so is there? And I've often, people are trying to pursue this with fMRI, to try to characterize the, when for instance you were talking about meditation in one of your papers, where people's attention, naturally sort of drifts. And is there is there a mechanism of that? Is there a carrier function of that? And you were also talking about, and it sort of touches on awareness versus not being aware. If you're aware that you're focused, if you're meditating, there's a lot of shifts that occur under your awareness as well. Once again, I'm sort of jumbling your papers together, but the story, I'm just trying to figure out, there's the alpha frequency, but then there's a modulation of that potentially. That's maybe another, I'm just wondering if there's sort of a timekeeper in the brain that just forces the attention to sort of drift naturally at a slower frequency as well.
That's an interesting thought, this latter point you made. I think it touches a little bit on the discussion in the cognitive sciences of flexibility versus stability, where the brain wants to be, able to consider new possibilities and doesn't want to be stuck in one state, but also wants to be able to focus on just one type of processing without all the time like going all over the place. And it seems to be that cognitively speaking, some people are better at one versus the
Yeah.
other, and there is some sort of a balance point.
Yes.
And so are you asking whether there are fluctuations in the brain that cause fluctuations in attention, or
Yeah. Maybe it probably could be some subcortical mechanism or something like that. But I'm wondering if there's something that could have a signature.
Yeah, and that's a big question, and I'm not very familiar with the literature of interventions such as meditation.
Yeah.
But breath focus, for example, which is a very typical type of meditation, or a beginning stage for other types of meditation that usually are chained after this first awareness on the breath, it does increase alpha frequency power. And so perhaps that would go hand in hand with reducing noise in the brain in some way.
Yeah.
Right. So that would be aligned. But I'm sure that if we delve deeper, and I'm sure the literature already has done quite a lot of that, we find more signatures that would be useful in terms of what to really enhance in neurofeedback kind of products. Of course,
Right.
Silicon Valley tries to sell you everything to train your focus, right?
Yeah.
Those products are only as good as the algorithm that is based on our current understanding of what actually drives certain states of focus in the
Yes.
brain.
Along those lines, potentially, have there been experiments? And I sort of briefly looked at the literature, and it looks like there has. Like, for instance, if you do TMS at 10 hertz, which is alpha frequency.
Yes. So, there is already really early work. I want to say maybe around 2009, 10. So first of all, before I mentioned the TMS, there were a couple of studies. One being here from Urbana-Champaign, Diane Beck's laboratory with Kyle Mathewson, I want to say, was the first author. So what they looked at is they had a perceptual task, and then they looked at the phase of the alpha band, and it looks like depending on whether the stimulus happened to fall in a certain part versus another part of the alpha phase,
Yep.
the participant was doing well in the perceptual task versus not doing well.
Yes.
And so that gives a sense of causality, but then the TMS papers came in, and Gregor Toot already did, again, around that time, already a 10 hertz TMS to show that depending on whether the stimulus falls at one part or the other part of this 10 hertz stimulation, you actually rhythmically manipulate perceptual abilities. So it actually provides causal proof for an impact of the alpha cycle on perceptual output.
Yes.
So that's older literature. I'm sure there is newer aspects in there.
Yeah.
Literature.
Yeah. And so, okay. So yeah, this is all, I mean, it's just, you have this sense that the information that you get with EEG or MEG is so rich, and it's just waiting to be explored exactly like you're doing right now. And what is the connection between, spiking and frequencies? Is it like some sort of,
chicken and egg, right? So that's, I'm glad you're bringing it up, because I've been really, let's say I'm surprised by the fact that in the year 2025, we still have a discussion of whether rhythms are epiphenomenal.
Yeah. Yes.
It's really mind-boggling to me, because that's the question essentially, as I'm hearing from you.
Yeah.
It's the question, if the spikes have a certain temporal pattern that aligns between a large enough population temporally, you're going to get rhythms, right? But do the rhythms have any information? If we knew every spike of the brain at every moment, okay, if we had that God view on the spiking, full
Yeah.
knowledge, would we know everything that there is? Or is there something about the rhythms that is emergent
Interesting.
that was not present in the totality of the spikes?
Yeah.
And
Yeah.
so there is different camps currently in electrophysiology, and there was a revived interest in aphatic coupling, which is the coupling of neurons beyond synaptic contact,
Okay. Okay.
be it chemical synaptic, like through neurotransmitter contacts, or even gap junctions, just coupling that's through the extracellular space.
Yes. Yes.
And there is rich empirical evidence that there is. The question is the effect size large enough to actually have any meaningful impact on how the brain functions?
Yeah.
To
Yeah.
me, it's obvious that the rhythms. So maybe this comes from my connectomics view, but I like to look at the brain at the whole. And I know, of course, that we lose an incredible amount of information when we look at the scale, right? But it seems to me that the large scale states of the brain are important for our conscious content, to get back to consciousness, or even non-conscious content. Everything that we do is dependent on the intrinsic brain states. And those intrinsic brain states, I don't think are well described, even if we knew every little spike,
Yeah.
would not completely describe the state when it is defined as a large scale population property, which is what the rhythms do. You don't get rhythms unless there is major cooperation or
Yes.
coupling of some sort. And that's why I brought up aphatic coupling. I would like to start thinking more broadly about electrical fields and magnetic fields. Magnetism is purely epiphenomenal in current neuroscience.
Yes.
And I just seem to think, if I were nature, why would I possibly evolve in such a way to, electromagnetism is one phenomenon. It
Yeah.
is not, but current neuroscience is only about electrochemical properties. So we're ignoring these fields.
Yeah.
And I
Yeah.
kind of sometimes feel like my students can't do this, but I'm really trying hard. I tell them, put yourself in where I was before I started grad school. Can you imagine a world in which people didn't know intrinsic activity was spatiotemporally organized? Because that is most of fMRI. That is the
Yeah.
world we grew up in.
Yeah.
They just can't even. They were like, I'm like, it wasn't the data. It wasn't even in the first ever fMRI recordings, right? Yes, they were single slice, but it was there all the time. We just chose not to look at it.
Right. Exactly.
By
Exactly.
the way we model, fMRI only gives you responses you're asking of it.
Yep.
If you don't model something, you won't see it.
You
It's
won't
not
see it.
there, right? And so I kind of feel we're somewhere there again, one more
Yeah.
time, which is very exciting. I
Yeah.
don't know what it is, but
Yeah.
it's in the data. I'm just trying to look for it. And I think thinking about the fields and stepping a little bit away from the individual spikes being the major code of the brain. I
Yep.
think that's anyway where I'm at the moment. And
Yeah.
so I think that would get us further. It didn't answer your question,
Yeah.
but
No, but it's I mean, it sort of broadened the nature of the question, which is perfect. Look at like Earl Miller at MIT. I mean, all he talks about is rhythms and it's really it just feels like he's just playing in this massive field of interesting possibilities of understanding how the rhythms interact.
the
And
field of fields.
yeah, yeah, and some I have some colleagues who are physicists. One person at UCSD, Larry Frank, who actually has this idea that aside from transmission along white matter tracks or synaptic transmission or dendritic transmission, he also, as I think you were implying, that there's connectivity on the surface of the scalp that might transfer information, or that not the scalp, but along the cortex that might, just simply the connectivity of the brain tissue might might be another source of
That's
communication.
right.
Um,
That's right.
so it's interesting. I mean, some people have tried to model. I have another friend UCSD, Eric Wong, who's a physicist, but he retired and now he's getting into sort of deep theoretical neuroscience, where you can model, like if you have a network of neurons that have some delay and they're sort of communicating and they're inhibitory in nature, you can actually produce different frequencies, alpha frequencies or whatever but it's very small scale. It's like just 10 neurons or something like that. And it's hard to imagine how that scales up in such a way that's coherent and produces that. But you're right. I mean, there's other mechanisms as well. There's other neurons in the brain. There's information that might be there that's not just in the spikes. I think that's what you're saying. But it's a wide open question, I feel. And it seems like, we're looking at these signals. And does it even make sense? I mean, you implied this in one of your papers that I think it was one of the review articles, that does it even really make sense? Certainly these frequencies come out and then we assign meaning to the frequencies.
That's right.
But does it make sense to break it into frequencies?
That's a really good question. I mean, it touches on the question of what the 1 over F overall spectrum is versus what true oscillatory rhythms are in the brain. And we talked a little bit about my EEG connectomics work and such, and we simply break the signal down into different speeds. And we use the canonical labels for those speeds, but I don't really think that they necessarily, especially in this, whole brain source space mesh that we're looking at, is reflective of those true rhythms that have been well described. Let's say when someone looks at hippocampal theta, they're looking at a very specific mechanism. If I now source localize to like a dense mesh from scalp EEG and project theta all over the brain, am I looking at the same mechanism? Maybe yes, maybe no, probably not.
Yeah.
I think a lot of the signal that we are looking at is different speeds of a larger scale-free signal. That doesn't mean we shouldn't break it into bands. We have actually one work under review right now, which shows that if you look at fMRI and concurrent EEG in the different frequency bands, and you look at how the canonical neurocognitive networks come and go, they are entirely unrelated to each other in terms of linear synchronization. There is something different happening at each one of these speeds. Yes. I like to call it multiplexing because it reminds on telecommunication systems. And you have one system, but you have different kind of information because you have different speeds at which things are happening. I tend to think of those as just different speeds of a larger scale-free signal. Whereas the alpha, of course, it really dances over the 1 over F spectrum. It's a true oscillation all across the brain. So I do not mechanistically imply that all of these are recurrent periodic rhythms.
Yeah. Okay. And actually, that's great that you brought that up. I was just about to mention that paper that you had. I guess it was from 2025. It just came out in eLife, actually, on the multiplex of connectome trajectories enables several connectivity patterns in parallel. And I thought that was really a cool paper. So basically the idea is that you have with fMRI, you have these, when you look at, windowed correlation or many other techniques, you can see the network sort of reconfiguring spontaneously. But then when you look at EEG and you have your approach to derive the connectivity networks, which in itself, I think more people should be using and embracing. I think it's really wonderful. Your paper implies that, like you just said, that there's all kinds of transitions that do rest in those spatial configurations, but the temporal sequence is all over the place. And it's very different than it doesn't just follow the fMRI.
That's right. So there was really one transition that happened in my brain forced by the data, not because I wanted to believe in it, but because nothing else made the data make any sense. My EEG fMRI trajectory started with the traditional thought that we should look for commonalities. If we look for commonalities, we get great spatial information from fMRI, we get temporal information from EEG, but that is not what the data says. It seems like there is, at least at the connectome level, there is a lot of temporally unaligned separate information in the fMRI that's not in the EEG bands and vice versa.
Yes.
And also not aligned between the different EEG frequency bands.
Yeah.
In fact,
Yeah.
the paper you brought up is really interesting to me because we were really fortunate to have intracranial EEG with fMRI in humans.
Okay.
Unheard of data.
Yeah.
I mean, just mind blowing. And this is through generous collaboration with Louis Lemieux and
Yes, I know Louis.
other colleagues. And so this data is from UCL. And obviously, every patient only has a part of the connectome. You know, they have their own little window into it, but it covers different lobes usually in most patients. And so when we look at the connectivity patterns in fMRI and concurrent invasive EEG, so this is not even an artifact of source leakage.
Yes.
Just look at them over time, and we look at them at all time pairs. So we slide one data modality over the other.
Yeah.
The idea would be that if similar connectivity patterns occur in the two data sets, and one is driving the other, or somehow they're related, then when we don't temporally slide them, when they're synchronized, there should be more similarity than when we disturb the timing.
Yes.
But that's not what we find. We find that there is spatially similar patterns in the two data modalities, but they don't occur at temporally related time points.
Huh.
Even if we HRF convolve the signal,
Right.
even if we account for the hemodynamic delay, it doesn't matter if we do or we do not. It does not change the fact that it seems like connectivity patterns in the EEG, even if we slow them down through the hemodynamic lens, right? They just don't align with fMRI at the connectome level. It doesn't mean that the fMRI connectivity at every edge does not have a physiological basis.
Yes.
So I'm not saying fMRI is not
No,
physiological, right? There's so much more spatial information in each data modality that perhaps comes from different neural processes.
Interesting.
And what always comes to my mind when I explain this finding, which the paper I mentioned under review finds the same thing with very different approaches. It always brings to mind the Talerac lecture of Rita Hari in Rome in 2019. It's online for people who weren't lucky to be at that particular meeting. I was very happy to be there because she spent an hour. The talk is called Timing Matters or something like that.
Yeah.
She spends this entire hour, with all this wealth of decades of knowledge she has from the birth of MEG to today.
Yes.
And its relationship to fMRI and why they're not lining up. It's all about, she's going so far as to say, and that's the way I hear the lecture, so you can go and check for yourself. But what I heard from her is that the neural processes, in terms of what fibers and what neurotransmitters are actually majorly contributing to fMRI versus those that contribute to EEG, MEG, they're not even the same. And so why are we even expecting this much alignment between the two data sets? Once we stepped away in my lab from expecting that alignment, a lot of things fell into place. Again, I'm not suggesting fMRI does not have any relationship. All of the alpha stuff that I explained was synchronized regression of one data modality on the other.
Yes.
It's certainly there. I'm not doubting it's there. But there's so much more.
Yeah. So my intuition would say, well, okay, there might be not much relationship, but it seems like statistically, you have to have more dwelling in a certain network to generate the fMRI signal. It seems.
Yes.
So a little bit more.
You definitely have those that might not be very dominant in the EEG, which is already a certain window onto electrophysiology, which may or may not align with the window that fMRI has onto electrophysiology. They might have different. Again, the reason I brought up Rita Hari's point is that I think what she was suggesting is that they may have different parts of the electrophysiology that they capture better.
Okay.
Just due to the nature of what they cover. And her major point was that cognition is by necessity multi-time scale.
Yep. Yeah.
And that is why the brain evolved to have both slow and fast processes, because the brain is reflecting the statistical regularities of the stimuli out there. She has this example of a tree moving in the wind, and the leaves with the lowest mass are vibrating really fast, and then the small branches and the large branches and the trunk and so on. And that's in vision. And audition, it's even more obvious. If you decompose my voice after this recording, you're going to obviously see that my brain, in synchronous time, needs to process various different frequency bands.
Yes.
And each of these processes involves distant brain regions that need to communicate with each other in order to separately look at prosody and my stress points, at the
Yep.
phonemes,
Yep.
and so on and so forth. And so if the brain already solves everything at multiple timescales in parallel, what other than multiplexing could the
Yeah.
brain possibly do?
Yeah. No, that's interesting. I love that sort of perspective. So your brain is sort of like, evolved to be tuned to all the evolutionarily relevant, stimuli in nature. And so it's, from very, very fast to very slow. And yeah. And so then what jumps to my mind then is that, you have all these computational cognitive neuroscientists who use fMRI and they try to model it. And it's like, okay, and I always sort of feel like, okay, you're modeling one scale that's like really slow and trying to derive some sort of mechanistic information about what the brain is doing. And is that really, I mean, it's certainly great work and I'm not going to, it's useful in itself, but it sounds like, if you really want to understand the brain, you have to go across scales a little bit more and trying to, get our heads around your data in terms of the EEG, to model that, it seems like would be much more challenging. But it's something. It feels like there's like the question is like where is, if you're trying to derive principles of like how the brain functions, is the high temporal scale the scale you really should be looking at as opposed to the slow scales. I mean, you can get information from that, but
Well, I feel that we need all of it. So
yeah,
the reason I feel that is I think it was a lucky strike in some ways that fMRI is slow.
yeah, yeah. It
I
integrates.
actually, exactly. For the same reasons I said, there's got to be something cognitively very slow and stable in terms of self memory of its immediate past and
Yeah,
longer term past that needs to be there in the brain. And I think fMRI just happens to be particularly good at overemphasizing those things that are stable over several seconds.
yes,
And I think it's lucky for cognitive neuroscience if we realize that this is cognitively important, but it's not just a slowed down version of the fast things that really matter.
yeah,
I
yeah,
think as cognitive neuroscience, we have to acknowledge that there are fast things that really matter to cognition, and there are slow things that really matter to cognition.
yes,
Not just because fMRI is just a dumb method and it just happens to be slow, you know? And so that I think that's the beauty of it. I really start appreciating fMRI beyond when I tell people I find stuff in EEG that's not an fMRI, people are like, I mean, obviously you'll find it because there's fast stuff fMRI can't see. And I'm like, yeah, but the reverse is also true.
yeah,
And
yeah,
that's not just because EEG is at the scalp level. If you go intracranially and you look at the aligned fMRI voxels and the specific electrodes, including depth, right? You still see that pattern. So I think there is slow things that inherently need to be slow because it's advantageous for the brain.
yes,
And you said something about principles, like how do we need to, at what scale do we need to model to find principles? I think if we also thought about what principles allow us to understand why certain things happen at fast and slow scales,
yep,
these are going to be scale overarching principles that have a different type of value. I think multi-time scale properties or at least scale freeness, if we find principles that repeat at different scales, scale freeness is a property of complex systems
yep, yeah,
such as the brain. And that brings me back to the point I made earlier, that I think that the fact that the brain is so ultra connected with features of complex systems such as small worldness
yeah, yeah,
makes it necessarily be imposing similar principles across time
yeah,
scales.
yeah.
And
That's
maybe we could find those and model them across scales, and it would be formula that repeat. And I think that there's something beautiful about it, which is, I think, one reason why physicists are so somehow, I don't know, they love complex networks and they love scale invariance. And, some neuroscientists do, and others are asking, why should I care about scale
yeah.
invariance? And it's like,
It's like some elegant principle that we
it happens
gravitate
to be
towards those
there.
things. Yeah,
Right. So I think it all kind of ties together. I think it's modeling fMRI as such is really valuable, and acknowledging that fMRI really is a good window on slow but very important things,
yeah,
but not just the slowed down version of something fast.
yeah. I agree with that. I just laying out the nature of the problem. Yeah, and I think and that brings me to one of I mean, it sort of relates to this in this sense. And so, and I don't usually I wasn't going to bring it up, but you had wrote a really nice review of Luis Pessoa's book, The Entangled Brain, and I recommend reading the book. I haven't read it yet, but I'd like to. But you mentioned, and actually this was brought up in other cases too. Nicole Rust has used this analogy in terms of how it'd be hard to sort of go into the circuitry and predict the brain. The idea of, but you used in your review, you wrote analogy of weather. There's a trade-off between coverage and spatial resolution, and we can predict the weather pretty well, but because it's a chaotic system, it's hard to predict beyond a certain amount unless we get very, very detailed information, and it's still very sensitive to initial conditions. And it's interesting. So you said, trade-off between coverage and spatial resolution yields a reasonable predictions at an acceptable computational cost. Like the mesoscale models provide the most accurate predictions when informed by parameters stemming from the global. So in other words, it might, I guess what I'm trying to say, in the brain, you can there's a sweet spot in some sense where you can get a lot of information from, not going all the way down to the single neurons and trying to look at how the systems interact. At least that's what I derived from your paragraph.
Yeah, I think so. Again, it relates to the question of whether if we looked at spiking hard enough or had enough spiking information, how much would we know? And I think if someone, for example, is interested in audition and they look at a whole lot of electrodes across a whole lot of layers of auditory system, would they know? Would they understand audition?
Yeah.
Or is there something missing?
Yep.
Right? The weather is a fun example because what happens here can't be modeled just by looking at the Midwest.
Right.
That'd be a pretty miserable model. It would not have predicted this recent snowstorm. So, I mean, there's this famous example of the butterfly fluttering somewhere. I don't know, was it in Brazil or whatever? And then so this is the nature of massively interconnected systems, where it's not like you get the global weather by the local weather and that's it. Right. The local weather is dictated by the global as well, but it's a loop.
Yes.
And so can you look at the brain just looking at all the local information? Imagine again, we had the full view of every spike. Would we expect we would know the full weather of the brain? And so Luis Pessoa's direction, as I understand it, is that no, you cannot.
Yeah.
Because the local and the global are so intertwined in both directions that you have a complex system where the total weather emerges as the total phenomenon. And he likes using the word emergent, although I think it's a hard, that needs to be defined, but it comes to the question of scale. How much can we understand about the brain without looking at a fairly large scale aspect? I mean, obviously, we can't do all the research at all levels at all the time. So we just have to have research that focuses on the small bits and research that focuses on the large bits. But we also need research that actually, at least for some process or some part of the brain, tries to bridge the two.
Yeah.
Otherwise, we have the local and we have the global, and we'll still not be able to model what the brain is actually doing, right? Or understand it.
Yeah, and I think you need the global. If for instance, one of the big hopes clinically, the NIH funds all these projects, and is the idea that you can actually, once you understand the circuitry enough, you can go in and intervene, affect some of the circuitry. And Nicole Rust mentioned that that's potentially problematic because, like you said, we don't know beyond a certain time window or spatial window. We don't know how that is influenced or is, because it's a incredibly multi-connected system, complex, it's hard to predict what will happen if you actually do that, if you try to intervene and change something. And so it was sort of a bleak picture in terms of how, if you can actually use this information clinically. If you had a perfect TMS probe that could go in or chemically or magnetically do that.
That's right. So the question of what scale is the most promising scale of intervention, because certain scales represent larger amounts of interaction. Right? Yeah. It's I mean, if I could answer that, I would probably get a lot of NIH funding.
Yeah, that's one of the big questions. But anyway, so we've already, time flies. We're already well beyond the hour. But so maybe this has been awesome conversation. Yeah, there's so much more we could talk about. Maybe we'll have to have you back again. But I just wanted to end up with, I usually end up the podcast with sort of like general perspectives on the future, advice to young scientists. It sounds like you kind of came the route through cognitive neuroscience, but you got a lot of skills and what you're doing. And I have to emphasize this: your approach is both, it's bold in a sense, and I think that you're defining your field and pushing the field because you're not afraid to sort of not follow the convention in terms of how to look at the EEG signals. And I think that's an important principle of how to do research. But what do you think? What advice would you give? What's to new researchers starting out who might be interested in this, or what skills do they have to know? How do they build collaborations? Because you have so many as well. I mean, how do you forge this sort of work further?
No, that's a big question. I think again, I was very fortunate. I don't think everything was by design. The connections that happened. But
Yeah.
you mentioned the word skill, and I think for some reason from the beginning, I was fortunate to get a lot of information to think more deeply about the sources of the signals that I'm looking at, and I think that's been very helpful. And so with the different people I've worked with, I've gained a lot of different tools and perspectives on how to analyze data, how to design experiments, but also again thinking about the deeper level of the signal. So I think I would definitely have the recommendation of not just focusing, let's say, on cognitive neuroscience without understanding the very basics and challenging the very basics of the signals. I'm getting new viewpoints onto the fMRI signal beyond what's traditionally thought to cause it, and it's very exciting. I think it's going to lead my research forward. So I think thinking deeply about what it is we're measuring alongside cognitive neuroscience, but also, if you're on the method side, on the other end, make connections to also get this relationship to behavior, and sort of try to do both in terms of the different maybe trajectories of laboratories or collaborators that you're selecting. But again, I don't think a lot of it was by design, and it brings one sentence of Mark Esposito to mind. When I was entering the job market as a postdoc, I was, nervous, as many people are. What are the chances I get an interview? So, and he told me something that really stuck, and that's totally Mark. He said, "Everything you've done so far has worked out. Just keep doing what you're doing." And I'm like, so just relax. So he seems the most laid back person I've come across. And so really, I think maybe I just give his recommendation forward to the other young scientists who are perhaps thinking about pursuing an academic career further.
Yeah, that's great advice. And I love Mark's approach because, and I kind of have it in my lab where people come in and they want to know what to do, and I'm like, well, I try to figure out what they like, and I try to figure out what resonates with them, and then have them go for it. Never really works typically from a mentor's view to just try to forge people into, okay, now we're going to do this specific question. Sometimes the grant funding structure forces that, but sometimes you're allowed some latitude. And I resonate with your perspective of understanding the source of the signal. The more you think deeply on that, the more you realize you can maybe separate the noise and actually derive new information that might be related to something from that. And that's so rich. This is what we do. We make measurements and we try to understand where they come from exactly. And the more deeply you can go with that, the more better questions you can ask. And that's what you're doing. That's great.
Thank you.
Well, this was great. This was a wonderful discussion, and I really appreciate spending the time. And I wish you the best of luck in the future.
This was a pleasure. How exciting. And thank you so much. And thank you for your service. I mean, these are amazing, amazing pieces. I really love listening to them. So thank you so much.
Oh, well, thank you.